Coming up on Drug Talk, a Drug Stories edition, we dive into the story of Alexander Fleming's discovery of penicillin. Contrary to popular belief, the story of penicillin is not one of just pure luck, but also a story of scientific perseverance, dedication, and keen scientific intuition. You will have the chance to be fascinated by hearing Alexander Fleming himself talk about penicillin, and we will explore some of the untold stories on penicillin's discovery. Who were the men that helped Fleming achieve his dream for penicillin? What is the connection of the famous D-Day with penicillin? And did you know that a cantaloupe melon played an important role in the use of penicillin in World War II? Coming up on Drug Talk. Welcome to Drug Talk, the remedy that brings you closer to the science of drugs and the stories that hold their secrets. Hello everyone, and welcome to the first episode of Drug Talk. This is the first of our 3-episode series of Drug Stories Edition, in which we will explore fascinating stories about the discovery of drugs. In this first episode, we are going to talk about the mother of all drug discoveries, Alexander Fleming's story of penicillin. Most of you should have heard the story, or should I say parts of the story, of how Fleming discovered penicillin by chance. But trust me, this is not the whole story. By the end of this episode, you will be convinced that Fleming's discovery was more than just a lucky one. It was a discovery based on teamwork and scientific perseverance. Alexander Fleming is famous for being the one that discovered the world's first widely effective antibiotic. This discovery revolutionized medicine and has been described as the single greatest victory ever achieved over disease. It's estimated that well over 200 million people have had their lives saved by penicillin in the first few decades of its use, and it continues to be an effective medicine used all around the world to this day. But first, who was Alexander Fleming? Alexander Fleming was born on the 6th of August, 1881 in Darvel, a small town in East Ayrshire in Scotland. He lived a relatively normal childhood until the age of 7 when his father sadly passed away. Luckily, his family made it work with his father not being there, and Alexander earned a scholarship to the Kilmarnock Academy, which then later enabled him to move to London, where he attended the Royal Polytechnic Institution in Westminster. From there, Alexander followed the career path of his brother Tom Fleming, who was a doctor. And in 1903, Alexander enrolled at St. Mary's Hospital Medical School in Paddington, London. Whilst at medical school, Alexander joined the Rifle Club and was reportedly a good shot and a vital member of the team. So once Fleming finished his degree and was set to leave the medical school and the Rifle Club, the captain of the Rifle Club, wishing to keep Fleming as a member of his team, suggested he join the research department at St. Mary's Hospital in order to stay one more year with them. At that time, in St. Mary's Hospital, a microbiologist named Almroth Edward Wright was trying to expand his new inoculation department. Wright was the real deal. He developed the first vaccine against typhoid fever in 1895, and his lab would be all about vaccines, antiseptics, and anything that could treat infectious disease. So in 1906, the 25-year-old Scottish research assistant Alexander Fleming joined the St. Mary's Research Department and became the assistant bacteriologist to Sir Almroth Wright. This was a key moment in Fleming's life, and quite frankly, for humanity. Joining this research department set him on a path to discovering penicillin and changing the world. Fleming spent his first few years at St. Mary's researching another antimicrobial called Salvarsan, an anti-syphilis drug, which came out in 1909. But then World War I kicked up, and some of the St. Mary's Hospital staff went over to France, where they did some really cool research on wound infections. At this point in medical history, germ theory is pretty well established, so most doctors would treat a battlefield wound with an antiseptic to prevent infection. Alexander Fleming served throughout the First World War in the Royal Army Medical Corps. During his time in the First World War, Alexander and Sir Wright moved the entire inoculation department of St. Mary's to the British military hospital at Boulogne-sur-Mer in France. While serving there, they witnessed the deaths of many soldiers from sepsis, a life-threatening condition where your body overreacts to an infection and starts attacking its own tissues and organs. Fleming observed that the antiseptics which were commonly used to treat infected wounds at the time often didn't work or even worsened the injuries. In an article he published in 1917 in The Lancet, a medical journal, he explained how antiseptics were actually killing more soldiers than the infections they were trying to treat. Antiseptics worked well on the surface, but deep wounds tended to shelter bacteria from the antiseptic agent. Antiseptics also appeared to remove beneficial substances created by the body to help fight the infection. And once these were removed, the bacteria the antiseptic failed to kill had free rein to infect the body. Alexander Fleming's mentor, Almroth Wright, strongly supported Fleming's findings. But despite this, most physicians working during the First World War continued to use antiseptics to treat infections, even in cases where it worsened the condition of the patient. After the First World War, Alexander Fleming went back to working at St. Mary's Hospital, where he investigated antibacterial substances and their effects on animal tissues. In November 1921, Fleming came to work with a cold, and so he did what anyone would do in that situation and put some of his drippy, mucousy snot on a petri dish. Now, I looked everywhere for documentation about why Fleming decided to do this and couldn't find anything, just that he did. So, as far as I can tell, Fleming was just following his curiosity at the moment. He was possibly exploring whether his mucus had any identifiable bacteria in it. He left out the dish with snot on it and came back to it 2 weeks later when, of course, a bunch of bacteria had grown on the dish, except for in the places where he put his mucus. Nasal mucus contains enzymes and antibodies as part of the body's first line of defense against pathogens. To his surprise, he found the mucus inhibited the growth of bacteria that grew in the agar plate, exhibiting by a clear, transparent circle on the agar plate, which indicated a killing zone of bacteria. Fleming then decided to test nasal mucus on a different type of bacteria that was growing in a yellow saline solution. Within 2 minutes of adding fresh nasal mucus, the yellow saline turned completely clear. Fleming then had the idea to experiment with tears rather than mucus, asking all of his co-workers to contribute. As Viddy Allison, one of Alexander Fleming's research colleagues, recalled, "For the next 5 or 6 weeks, our tears were the source of supply for this extraordinary phenomenon. Many lemons were used after the failure of onions to produce a flow of tears. The demand by us for tears was so great that laboratory attendants were pressed into service, receiving 3 points for each contribution." Alexander Fleming went on to not just test mucus and tears, but also other bodily samples, such as sputum, cartilage, blood, semen, ovarian cysts fluid, pus, and egg white, proving that the antimicrobial enzyme lysozyme that inhibited bacterial growth was present in all of these bodily samples. Fleming reported his new discovery to the Medical Research Club and Royal Society, but no one seemed that interested. His paper describing lysozyme received no questions, no discussion, which was a pretty clear indication that people viewed it as insignificant. Lysozyme had very little therapeutic potential, as the enzyme was only really effective against small amounts of relatively harmless bacteria, but it was still an important discovery that helped to understand how animals' immune systems, including our own, worked. It wasn't until the late 20th century that the importance of Fleming's discovery of lysozyme was recognized. It was actually the first antimicrobial protein ever discovered, made up part of our innate immune system. After Fleming's discovery of lysozyme, he continued to study bacteria for many years in his laboratory at St. Mary's Hospital. In September of 1928, Fleming was going on holiday with his family to Suffolk. Before leaving his laboratory, he inoculated Staphylococcus bacteria on culture plates and left them in a corner of his lab. Vidi Allison recalled that Fleming was not a tidy researcher, and as a result would regularly expect unusual bacterial growths to appear in his culture plates. But this untidiness, at least in this case, was probably a good thing. On Fleming's return from his holiday on the 28th of September 1928, he found that one of the culture plates he had inoculated was contaminated with a fungus, and the Staphylococcus bacteria immediately surrounding the fungus had been destroyed, presenting a clear killing zone similar to the one observed in the earlier case of lysozyme. Upon seeing this, Fleming famously remarked for this coincidence, "That's funny." Alexander Fleming identified the mold growing on the culture plate as being from the genus Penicillium. He went on to grow more of this mold and quickly discovered it contained antibacterial properties. After further investigations, Fleming found it had a pronounced impact upon Staphylococcus bacteria and other similar gram-positive pathogens that can cause things like scarlet fever, pneumonia, meningitis, and diphtheria. This accidental discovery would turn out to be one of the most significant medical breakthroughs in human history. Little did Alexander Fleming know at the time, but he had just discovered something that would go on to save hundreds of millions of lives and totally change the world. Nevertheless, his earlier experience with lysozyme's antibacterial activity had most likely played a crucial role in Fleming's attention to what he was seeing in his Staphylococcus agar plate. Fleming initially just called what he had discovered mold juice, but after several months of work, he gave the antibacterial substance present in the mold the name penicillin, which is still known as today. It's important to mention here that although Fleming first identified and named the mold's antibacterial penicillin, he wasn't the first to notice penicillium's antibacterial effects. There are texts that mention the ancient Egyptians used molds against infections, millennia before Fleming. And even more recent scientists like John Tyndall described antibacterial effects of a mold in 1876, more than 50 years earlier than Fleming. However, germ theory was not much supported back then, and Fleming's predecessors didn't progress as to identify a single substance in the mold having the antibacterial property. Alexander Fleming presented his new discovery to the Medical Research Club in 1929, but once again, nobody seemed that interested. The chair of the meeting later reminisced that he didn't sense any importance in Fleming's discovery at the time. Fleming went on to publish his work in the British Journal of Experimental Pathology, but once again, little attention was paid to the article. This reaction had a true basis. Fleming and his assistant never managed to purify penicillin very well. They couldn't get it higher than a 1% concentration, and penicillin was tricky to work with. It would evaporate and leave a sticky, ineffective syrup. And while they did test penicillin for safety in lab animals, they never tried to treat bacterial infections in animals. The difficulty of isolating the main compound of penicillin and producing large amounts of it was one of the main reasons for a lack of interest. If production couldn't be scaled up, penicillin was deemed effectively useless to the medical community. Fleming later received help from Howard Rainstick and his team of biochemists from the London School of Hygiene and Tropical Medicine, but they came to the conclusion that the chemical purification of penicillin was futile. They just thought it could not be done. As a result, penicillin was largely forgotten about throughout the 1930s, although it's worth noting Alexander Fleming did not give up. He always knew penicillin's potential. Fleming first tried to use penicillin clinically in 1929 on a research scholar named Stuart Craddock, who developed a severe infection of his nasal cavity. Weirdly enough, the penicillin didn't seem to have any effect. Fleming thought this was likely because the infection was caused by a type of bacteria that was resistant to penicillin. The first recorded successful use of penicillin was not actually conducted by Alexander Fleming, but by Cecil George Payne, who was a pathologist and former student of Fleming. Payne successfully used penicillin in 1930 to treat 1 adult and 3 infants with conjunctivitis eye infection. It wasn't until 1932 that Fleming himself first successfully treated someone with penicillin. It was Keith Bernard Rogers, a medical student at St. Mary's Hospital and captain of the university rifle team at the time. Rogers developed a severe conjunctivitis eye infection. He had planned on participating in a rifle shooting competition, but due to his eye infection, was going to pull out. However, Fleming applied penicillin ointment to Rogers' eye, curing his conjunctivitis, and it's even reported that Rogers went on to actually win the shooting competition, all thanks to Fleming's penicillin. In 1936, at the Second International Congress of Microbiology, Alexander Fleming gave presentations about penicillin's importance and potential value for the treatment of infections, but once again, no one really listened to him. Even when he had the opportunity to present a paper of his work on penicillin at that meeting, which was attended by the most prominent bacteriologists from all over the world, they failed to see any value in his work. Fleming's core idea was that penicillin could be used to prevent and treat bacterial infections, but many of the bacteriologists there simply did not support this view. But why was there a complete lack of interest or discussion about penicillin by his peers? Many scientists remained skeptical at the time due to the novel concept of using mold-derived substances as antibiotics. It seems that they allowed their bias to affect their scientific judgment for the evidence itself, provided by Fleming's experiments. Additionally, technical challenges in purifying and producing penicillin in effective quantities further fueled skepticism, whether this was worth pursuing further. The scientific community was cautious, accustomed to more immediate and observable results from traditional treatments, leaving Fleming's revolutionary ideas unappreciated for some time. Moreover, by 1935, the German company IG Farben came out with sulfanilamide, another antibacterial against germs like Staphylococcus and Streptococcus, the same pathogens Fleming tested. And so, the penicillin research sat on the shelf for years. As a result of his peers' indifference, Alexander Fleming was utterly deflated. His fellow bacteriologists from all over the world, people who had dedicated their lives to studying bacteria just as he had, could not see any value in his work. It must have been so disappointing for Fleming. However, even this setback did not dissuade him from continuing to work on, to investigate, and to experiment with penicillin. He was convinced of penicillin's potential medical value to the world and was determined to make it a reality. Looking back now, it really is staggering how almost no one during the 1930s besides Alexander Fleming and some of his closest colleagues saw the potential of penicillin. Without Fleming's perseverance, penicillin may never have become a widely available drug. Alexander's conviction and determination was a real gift to humanity. The millions upon millions of people who have had their lives saved by penicillin owe a great debt to his dedication to the importance of scientific evidence. 9 years passed, and in 1939, Fleming's perseverance paid off. Howard Florey, an Australian who spent most of his career in the UK, became the director of the Dunn School of Pathology at the University of Oxford. One of his first hires was Ernst Chain, a Jewish biochemist who grew up in Berlin, but became an English citizen in 1933, right as the Nazis were taking over Germany. Florey and Chain start thinking about a new project. They wanted to do a big survey of the antibacterial properties of different substances made by microbes. So they started their project with a literature search, like any scientist should. And in 1937, Chain came across Fleming's original paper about penicillin. This gave the team the idea that penicillin might be a special subtype of lysozyme. The team at Oxford worked to produce a concentrated extract of penicillin that could be used in a powdered form. They started making more and more penicillin, and they were learning more and more about how it worked, but there was a problem. They were nearly out of money. Florey was able to get a little bit of cash from the UK's Medical Research Council, but he knew he needed more. so he applied for a grant from the Rockefeller Foundation, which he got by March 1940, receiving $25,000, which was a lot of money back then. The team conducted their first in vivo study of penicillin by injecting 8 mice with a virulent strain of bacteria, then injecting 4 of those mice with penicillin extract, leaving the other 4 untreated for comparison. After just 1 day, the 4 mice that didn't receive any penicillin were dead, whereas the 4 that did receive penicillin survived. Ernest Chain's remark upon seeing the result was immediate. He said, "It's a miracle." Florey and the Oxford team didn't think at the time that they're going to revolutionize medicine. Actually, quite the opposite. Florey is quoted as saying, "I don't think that the idea of helping the suffering humanity ever entered our minds." The team published their first findings in the Lancet Journal in 1940, at the beginning of World War II, entitled "Penicillin as a Chemotherapeutic Agent," and made it clear they had something new and useful on their hands. It's worth noting that Fleming was not in the authorship of that paper. Shortly after publication, Alexander Fleming telephoned Howard Florey to say that he would be visiting the team at Oxford within the next few days. When Florey told his team that Fleming was coming to visit, Ernest Chain famously said, "Good God, I thought he was dead." In 1941, the Oxford team attempted to treat with purified penicillin their first human patient. They gave some to a junior doctor named Charles Fletcher, who worked at the infirmary at Oxford. And on January 17, 1941, a woman with terminal cancer agreed to try it out. They weren't trying to cure her cancer, just seeing if she'd tolerate the antibiotic. Their first shot was an injection of 100 milligrams of penicillin, and it did not go well at first. While the lab had figured out methods for increasing the concentration of penicillin in the mold, they had also increased the amount of contaminants. This first injection was less than 5% pure penicillin, and the volunteer developed a fever and what they called rigor. Fortunately, she tolerated the second injection much better. Their first test for effectiveness came about a month later. A few months prior, a 48-year-old policeman named Albert Alexander cut himself while doing yard work, and over the next few months had developed a life-threatening infection with both staph and strep bacteria. He was admitted to the infirmary, where he was treated with sulfa drugs, but the infection kept spreading. So on February 12, 1941, he received an injection of 200 milligrams of 5% penicillin and continued to get injections over the next 5 days. And for those 5 days, it seemed to be working. This was, to quote Fletcher, "the nearest I ever came to seeing a miracle." Unfortunately, there wasn't enough penicillin to finish the treatment. They even collected and tried recycling penicillin from the man's urine, but they finally ran out, and the man succumbed to the infection and died on March 15, 1941. Not a great start for penicillin, but this first use did teach the Oxford team a valuable lesson. For penicillin to be effective in treating severe infections, it needed to be given in much larger quantities. A year later, in 1942, Alexander Fleming carried out an important clinical trial. Harry Lambert, who was a colleague of Alexander Fleming's brother, was suffering from a potentially fatal infection of his nervous system. Fleming asked the team at Oxford for a purified sample of penicillin, which he then used to inject into Harry Lambert's spinal canal. Within just a day, Lambert started showing signs of improvement, and within a week, he had recovered. Fleming reported this clinical trial in The Lancet in 1943, and this evidence would later prove vital in helping to convince governments to mass-produce penicillin. I think it's important to stop for a second and realize just how monumental this moment was for humanity. Before penicillin, contracting a bacterial infection had the potential to be life-threatening. At that time, 90% of children with bacterial meningitis died. Common problems like strep throat and ear infections could spread throughout the body and cause serious medical issues. Alexander Fleming's discovery of penicillin, and to be fair, Howard Florey's and Ernest Chain's work in its purification changed all of that. For the first time in human history, we had a powerful and effective drug to fight bacterial infections. It opened the door to the mass rollout of antibiotics and completely changed our world. The timing was also important in penicillin's story. In the 1940s, the Second World War was raging fast. As with all previous wars, infected wounds were a huge problem in war. A mild cut or injury acquired on the battlefield had the potential to become infected and turn into a life-threatening issue for thousands of soldiers. Penicillin offered a credible solution to this problem, and it would be a real advantage in the war to have an effective antibiotic to treat the troops. However, there was an issue. Thousands of infected soldiers meant that the production of penicillin had to be seriously scaled up. Initially, in 1941, the Oxford team tried to convince the British government to mass-produce penicillin. But at that time, they just couldn't persuade them. Most likely because of the need of valuable industrial resources for the war that could not be spared for a risky project. So, Howard Florey and other members of the Oxford team traveled to the U.S. to persuade the American government and the American pharmaceutical companies to help mass-produce penicillin. They met with a number of government and pharmaceutical officials who immediately saw the potential of penicillin. Work quickly began at a USDA laboratory in Peoria, Illinois, and by the end of the first month, they were making their own culture of penicillin. However, they realized Alexander Fleming's original mold sample that all previous cultures had been grown from was not efficient enough to produce large quantities of penicillin. So, with the help of the U.S. Army Transport Command, a great search began to find similar but more efficient mold samples from all around the world. They brought back samples from China, India, South Africa, but the single best sample was actually from a cantaloupe melon purchased in a fruit market in Illinois near the laboratory by their lab assistant, Mary Hunt. When she brought it back to the lab, it turned out this strain had 200 times the penicillin compared to Fleming's Penicillium. After radiation treatment, they eventually got a mold that produced 1,000 times more penicillin than the original mold. Fun fact, that mold strain of that cantaloupe melon is the source for most penicillin in the world today. Between 1941 and 1943, researchers in America focused on industrializing penicillin production and isolating higher-yielding strains of penicillin. Simultaneously, researchers working for Pfizer, a pharmaceutical company, developed a practical deep-tank fermentation method that would enable large quantities of pharmaceutical-grade penicillin to be produced. At this point, penicillin had proven to be effective, and there were realistic methods for mass production, so the British Ministry of Health was contacted, convinced of the importance of penicillin. The British War Cabinet was convened and also convinced of penicillin's potential. And, as a result, the Penicillin Committee was formed in April of 1943. On the committee was Alexander Fleming, Howard Florey, and representatives from the pharmaceutical industry. The main goal of the committee was to facilitate the production of large quantities of penicillin as fast as possible. And they planned to do this in collaboration with American pharmaceutical companies, who were realistically the only people at the time capable of ramping up production. The Penicillin Committee aimed to exclusively supply Allied armed forces with the drug in an effort to help win the Second World War. And by June 6, 1944, otherwise known as D-Day, enough penicillin had been produced to treat all the wounded troops of Allied forces. And so, the world's first effective antibiotic, penicillin, became available to fight bacterial infections, a huge victory for humanity and modern medicine. And it didn't take long for penicillin to become available to the public. A typical advert from 1944 reads, "Penicillin cures gonorrhea in 4 hours. See your doctor today." After the Second World War had ended, penicillin was rolled out on a massive scale and immediately began saving lives. Doctors who had previously had few options in the fight against bacterial infections now had a powerful medicine. Truly, a momentous moment for humanity and modern medicine. Fleming's discovery and the hard work of the people of Oxford and the United States had changed the world forever. Alexander Fleming's discovery of penicillin earned him a long list of prestigious awards and titles. In 1943, he was elected a Fellow of the Royal Society. In 1944, he was knighted, becoming Sir Alexander Fleming. He was awarded the Medal of Merit by the President of the United States, was made a Grand Cross of the Legion of Honor by the French Republic, and was made Grand Cross of the Order of the Phoenix of Greece since his second wife, Amalia Koutsouri-Vourekas, later Amalia Fleming, was a colleague working with Alexander at St. Mary's. In 1945, Alexander Fleming, along with Howard Florey and Ernest Chain, shared the Nobel Prize in Physiology or Medicine for the discovery of penicillin and its therapeutic effects. In 1954, 25 years after Fleming's paper on his discovery, the St. Mary's Medical School in London celebrated this milestone with an extraordinary event in the presence of the Duke of Edinburgh, Prince Philip at the time. In his speech, Sir Alexander Fleming was gracious regarding the honors, but he made sure that the ultimate respect was paid to his mentor and guide, Sir Almroth Wright. Listen to Alexander Fleming's voice itself from an extraordinary preserved media material of that event in St. Mary's Medical School.
Your Royal Highness, my lords, ladies and gentlemen, I thank you, sir, for the kind things you said, and I've got to thank my colleagues for the wonderful tureens which I've just been presented with on this, the 25th anniversary of the publication of penicillin. It was just 25 years ago that the word first appeared in print. It was a brand new word, and people keep on asking me why I ever christened anything penicillin. Well, if you knew a little botany, you'd know, because the substance is made by a penicillium, so it's perfectly obvious to call it penicillin. Something happened which eventually led to penicillin, but would I ever have noticed it but for the previous work done in association with my teacher and master, Almroth Wright. See, when the First War came, we worked together in Boulogne, and we saw thousands of septic wounds that we could do very little for. The work which we did then in Boulogne and in the laboratories here was good work, I think. It's work I'm proud to have been associated with, and it made me especially interested in substances that killed microbes. So I noticed something that perhaps others had missed, and that observation 25 years ago has brought us all together today. See, I made the observation, but I don't forget what I owe to my teacher, Sir Almroth Wright, perhaps the greatest man ever to have been associated with St. Mary's Hospital Medical School in its 100 years of existence.
Alexander Fleming and the members of the Oxford team did not patent penicillin, as they thought doing so would be unethical. However, that didn't stop American pharmaceutical researchers from patenting the production of penicillin. And when Alexander Fleming discovered what they had done, he said, "I found penicillin and have given it free for the benefit of humanity. Why should it become a profit-making monopoly of manufacturers in another country?" It's easy to understand Fleming's frustration. He'd worked so hard discovering penicillin, not to make money, but to help humanity. And here were individuals in America trying to get rich from the hard work of Alexander Fleming and the team at Oxford. On the 11th of March, 1955, at age 73, Alexander Fleming died at his home in London of a heart attack. His ashes are buried in the crypt of St. Paul's Cathedral in London. But his death isn't the end of this story. Fleming's discovery of penicillin ushered in a new age of medicine. There are now over 100 different types of antibiotics available to help treat minor and life-threatening bacterial infections. Hundreds of millions of people have had their lives saved by penicillin, and it has been estimated that antibiotics have increased average life expectancy in the developed world by as much as 20 years. Today, antibiotics have become so widespread and easily available, even in the developing world, that it's hard to imagine life without them. They are crucial to so many aspects of modern healthcare. Every year, millions of operations are carried out, and antibiotics are used to prevent pre- and post-operative infections. 1 in 4 births are by cesarean, and antibiotics are used to protect both mother and child from infection. 50% of all women will suffer a urinary tract infection at some point in their life, and most of them will need antibiotics to treat the infection. Cancer treatments can suppress the body's immune system, so antibiotics are needed to help keep people alive whilst they receive their cancer care. And I could list 100 other examples of how antibiotics are now vital to modern healthcare. But here's the scary thing. We may soon be facing a reality where antibiotics no longer work. This is because of the rise of antibiotic resistance. Antibiotic resistance is a consequence of evolution by natural selection. Bacteria that cause infections would naturally develop resistance over time. But when antibiotics are used, it creates an environmental pressure on the bacteria, wiping out bacteria that are susceptible to antibiotics, but leaving the few bacteria that have a resistant mutation alive. These mutant bacteria live on to reproduce, passing their resistant mutation onto their offspring, which in turn creates a resistant generation of bacteria. This is why we need to avert abuse of antibiotics, promote correct use for the right conditions, and keep trying to improve our antibiotics as bacteria increase their resistance. Although the development of new antibiotics has been dramatically declining for decades, in the last few years, innovation and research around antibiotics have slowly begun to ramp up again. We still have a long way to go to where we need to be, but progress is being made in the right direction. To conclude this episode, Alexander Fleming discovered penicillin, giving humanity a powerful, world-changing medicine. But on his own, Fleming would never have brought penicillin to the fascinating medicine that has become. It required a number of skillful, resilient, and great scientists to work together, and with Fleming, to bring penicillin to the world. It also required political will and contribution of political and military personnel. Nevertheless, without Fleming's perseverance and vision, we may never have developed penicillin in the first place. As a direct result of Fleming's discovery, of Howard Florey's and Ernest Chain's hard work, and the contribution of a cantaloupe melon, hundreds of millions of people have had their lives saved and still have. Thank you for listening to this episode of Drug Talk, the first of a 3-episode series on drug stories, produced and edited by Dr. Nicholas Dietis. In our next episode, we will embark on a fascinating journey to explore the stories behind the discovery of warfarin, a drug that began as a rat poison but ended up saving the lives of millions of people, including the life of a U.S. president. Until next time, stay curious about the science that heals us.